Just to give you a heads up and a warning, apparently some of the other professors have had some issues with Collaborate today, so worst comes to worst, I'll use the morning lecture if we get cut off like we did that one time for lab, okay? But hopefully we'll get through this. Let's jump right into this. I'm going to just complete chapter 9, okay? I'll begin chapter 10 next week Monday because we have a little bit more time. So we were talking about cellular respiration. Talked about how it's considered to be consist of or fall under aerobic metabolism. We talked about how it's divided into 3 stages. Each stage consists of a series of chemical reactions catalyzed by a group of enzymes. We started by talking about stage 1 called glycolysis. When you look at the formula for cellular respiration, there are 2 reactants that are required, glucose and O2. The glucose specifically, I said, serves as a substrate. It's a substrate we need to begin glycolysis. Okay. O2 is not a substrate, but it is a reactant. However, I described it as being the final electron receptor. We'll talk about it today in stage 3. So that formula I give you for cellular respiration, it's more of a summary. It's not comparable to an actual chemical reaction. Now, we talked about how when we go over this process and when I test you on cellular respiration, nutrient is this form of chemical energy called glucose, and we're tracking, okay, how much ATP can be produced from 1 molecule of glucose. So we talked about how when glucose is used as the initial substrate for cellular respiration, it enters glycolysis, where a series of enzymes catalyze chemical reactions that results in the breaking of this 6-carbon backbone glucose into 2 3-carbon backbone molecules called pyruvate. So pyruvate is a final product. Okay. What was the first enzyme that catalyzed the first chemical reaction that acts on glucose as a substrate? What was the name of that enzyme? Okay, there are 9 of you here now. Come on, collectively, somebody will be able to give me an answer. So when glycolysis begins, it consists of 10 chemical reactions. What was the name of the enzyme that catalyzes the first chemical reaction and requires glucose as a substrate? It's tied into the shape of glucose. All right, so apparently my morning class wasn't just sleep. Hexokinase, okay? Hexokinase. Well, it's a series of 10 chemical reactions, so you also had to know the last enzyme that catalyzed the last chemical reaction that produced the products, pyruvate. What was the name of that last enzyme? Its name is tied into the substrate that it produces. Yeah, pyruvate kinase, okay? So these are the final products, and we said these 2 molecules of pyruvate that have a 3-carbon backbone derived from glucose would then be transported into the mitochondria, and an intermediate step would happen. It's not part of stage 1 glycolysis, it's not part of stage 2, the citric acid cycle. It's an intermediate step known as pyruvate oxidation, okay? These 2 molecules of these 2 molecules of pyruvate derived from glucose are both converted to acetyl-CoA, okay? So we end up with 2 molecules of acetyl-CoA. What do they mean by oxidation? Pyruvate oxidation. What was happened to pyruvate in the beginning when it entered this intermediate step? What was it losing? It was losing something that caused it to release one of the 3 atoms of carbon that formed its backbone, CO2. What was it losing that caused it to lose carbons as CO2? Right, electrons. Okay, so pyruvate is being oxidized, right? That's what we mean by oxidation. Any molecule that loses electrons, it's being oxidized. Any molecule that gains electrons, we refer to it. If a molecule gains electrons, if a molecule gains electrons, yeah, it's reduced. Okay. All right, now, this intermediate step, okay, the primary reason or purpose of this intermediate step is because of the citric acid cycle. Okay, again, just like with glycolysis, the citric acid cycle consists of a series of chemical reactions catalyzed by a group of enzymes. However, there's no true product ever produced. The purpose of the citric acid cycle is to simply complete the breakdown or oxidation of glucose, or in this case, glucose-derived molecules. Pyruvate and acetyl-CoA, these are molecules that came from what was once glucose, so those are examples of glucose-derived molecules. So, citric acid cycle completes energy-yielding oxidation of organic molecules. In other words, it completes the process of stripping as many electrons from glucose-derived products to the point that there is no product left. Okay, but this is going to result in the production of a little CO2. You're going to have to know two enzymes involved in the citric acid cycle. One is going to be the first enzyme that catalyzes the first chemical reaction of the citric acid cycle. That is the enzyme that doesn't recognize pyruvate as substrate, it recognizes acetyl-CoA. However, just like glycolysis, the purpose of the citric acid cycle, the primary purpose, is to extract electrons. Strip as many electrons from the glucose-derived products, or acetyl-CoA, as possible, except even more electrons are going to be stripped than what occurred in glycolysis. Now, I did it this morning. Let's see if I can do it again. Citric acid cycle. I was looking at a video link that I gave for my non-majors, and I believe I gave it for y'all, hopefully. But in that video, they don't use citric acid cycle. Citric acid cycle has two alternative names. Okay. I'm not looking at punctuation, I was writing it out. One is called the Krebs cycle. I'm not going to write out cycle, I'm just going to put C. The other one is a long name, tricarboxylic acid cycle, but as they often do in the sciences, long name, but they'll use abbreviations. So just to let you know, the citric acid cycle is also known as the Krebs cycle or the TCA cycle. Citric acid and Krebs are used the most between the two. Your textbook uses citric acid, that's why I use that, okay, everybody? So just to let you know. So if you watch one of the video links I put up, if they happen to say Krebs cycle, they're talking citric acid cycle, just to let you know. TCA, you don't hear as much, but if you do, know that they're talking citric acid cycle, stage 2 of cellular respiration. Okay, since there are a lot more electrons being stripped, okay, from this glucose-derived product called acetyl-CoA that enters the citric acid cycle, all right, it's not going to require one electron carrier, it's going to require two. In their oxidized form, we write these two electron carriers as NAD plus and FAD. I don't know why that equal sign is there. Doesn't need to be there. So down here, they're showing you here and here. There are a higher number of electrons that are going to be stripped from this glucose-derived product, acetyl-CoA. That is a substrate needed to get the citric acid cycle started. Okay. In their oxidized form, we write them as NAD plus and FAD, which is rewritten down here. The FAD, again, if anybody asks, that's what it stands for. I'm not going to test it on it, but there it is. But just like with NAD plus, the oxidized form of this electron carrier, I told you NAD plus can carry up to how many electrons? 2. And I said whenever NAD plus enters any part of cellular respiration, it'll enter in an oxidized form, meaning it's not carrying electrons. It'll come in and pick up 2 electrons, and it'll leave carrying 2 electrons. How do biochemists let you know whether it's in an oxidized or reduced form? They'll write it in this form if it's oxidized, not carrying 2 electrons. But when it picks up 2 electrons, when it gains 2 electrons, they will rewrite that electron carrier this way. This lets you know it's now in what form. What form is that again when we write it down as NADH, everybody? Right, the reduced form. Except, since so many more electrons are being stripped this time around, we have a second electronic carrier. In its oxidized form, it's called FAD. Okay, this electron carrier, just like the first type of electron carrier, can carry 2 electrons. So when it's not carrying 2 electrons, or when it's lost 2, we write it as FAD. When it enters the citric acid cycle and then leaves the citric acid cycle, it's picking up 2 electrons or gaining 2 electrons. Therefore, it's being reduced, and we write it this way to let someone know that it's now reduced, that it's now carrying 2 electrons, FADH2. Now, is there more to the story? Yes. Biochem fills it in. That's why you see these little guys like protons. Don't worry about that. This is what I want you to know. So now we have 2 types of electron carriers. But they also show you, just like with glycolysis, we're going to get a little ATP produced. Let me ask you this. In glycolysis, what was the total number of ATPs that were produced? What was the total number of molecules of ATP produced? Not the net, but the total for glycolysis. What, all of a sudden everybody disappeared? Again, what was the total number? Don't want to look at the chart. Okay, if you go back to glycolysis, 4 molecules of ATP total were produced. The problem is, or not the problem, but the issue is, it required how many molecules of ATP to help perform glycolysis? Does anybody know that? Yeah, 2. So with glycolysis, we talk about a net gain of 2 ATPs. 4 are produced, but 2 are required. So the cell ends up ahead by only 2. What I'm getting at is for the citric acid cycle, there's no ATP required by any of the enzymes to do their work. So ATP is not 100% of the time needed. Is that clear to everybody? Okay, it's just the most common source of, if you want to call it, fuel needed by proteins that do need it. However, what they're trying to show you here is for every 1 molecule of acetyl-CoA that enters the citric acid cycle and that is oxidized by having many electrons taken from it or losing electrons, for every 1 molecule of acetyl-CoA that enters the citric acid cycle, we get an equal number of molecules of ATP. Therefore, what is the yield per enters the citric acid cycle? Okay, we end up with a total of 3 electron carriers known as NAD plus being reduced to NADH. So in other words, 3 of them, okay, gain 2 electrons each. We end up with 1 electron carrier, FAD, entering the citric acid cycle, and it gains 2 electrons. So twice as many electrons have been stripped, and for each of these electron carriers, just like with glycolysis, just like with the intermediate step, what stage are these electron carriers in the citric acid cycle? What stage are they transferring or carrying these electrons to, everyone? Just the stage number. What stage? Right, stage 3. Yeah, they're all taking them to stage 3. So we'll explain why. Why is it when the electron carrier in its reduced form, NADH, exits glycolysis, why is it going to stage 3? Why is it the electron carrier in its reduced form, NADH, exiting the intermediate step and going to stage 3? And why is it both of these electron carriers are exiting the citric acid cycle in their reduced forms going to stage 3? We'll talk about that when we get there. Again, for every 1 molecule of acetyl-CoA, we get 1 molecule of ATP. But notice, no ATP is required. Okay, so we don't talk about net gain, we just get a gain for every molecule of acetyl-CoA. Also, for every molecule of acetyl-CoA, we get 2 molecules of CO2. So if you look at the intermediate step and you look at the citric acid cycle, by the end of the citric acid cycle, and when you include the intermediate step, you should be able to account for all 6 molecules of CO2 that are produced, okay, when 1 molecule of glucose is used for cellular respiration. So in other words, that formula, you should be able to add up some of the numbers is what I'm getting at. But the thing is, we're not tracking how much ATP is produced off 1 molecule of acetyl-CoA. I said we're tracking how much ATP is produced off of 1 molecule of glucose. This is the abbreviation for glucose, everybody. It's uppercase GLC. Well, we already talked about how in glycolysis, when electrons are taken from glucose, that breaks the 6-carbon backbone into 2 3-carbon backbones of pyruvate. Now, I'm just going to put PYR for pyruvate. Or I'm going to try to put PYR for pyruvate. Then we talked about how these molecules of pyruvate are then transported to the mitochondria, where they're converted to acetyl-CoA. I'm just going to write ACET, or I'm going to try to write ACET. So what I'm getting at is we get 1 molecule of ATP produced for each molecule of acetyl-CoA that enters the citric acid cycle. But if we're tracking how much ATP is being produced from 1 molecule of glucose, we don't want to just go by a 1-to-1 ratio, okay? In order for the number of ATP molecules that I give you in the formula, in order for that number, okay, to be reached, approximately 30 ATP, or 30 molecules of ATP can be produced from 1 molecule of glucose. What I'm saying is we have to take into account not 1 molecule of acetyl-CoA will enter the cycle, but tracking 1 molecule of glucose, 2 of these will enter the cycle. So simply put, what's the total number of ATP that is produced now for the citric acid cycle? We had a net gain of 2 for glycolysis. How many ATPs are produced for the citric acid cycle that the cell gains? If I'm making any sense to you. If you're tracking 1 glucose molecule. Process is happening very fast. As far as time goes, I don't know. Okay, I don't know in fractions of seconds, milliseconds is usually what we describe. So I don't know where it would fall in that range. Okay, I do know that the process can speed up and slow down, but it's still happening at a pretty rapid rate. I'm assuming it's in the milliseconds or sub-millisecond range. It just depends on the energy demand for the cell, but it's always happening unless the cell's dead. Okay, does that help out a little bit, Contreras? Because he was asking me how fast does the process happen. Okay, so listen carefully. What I'm getting at is we get a net gain of 2 molecules in glycolysis, and if we're tracking how much ATP we're getting out of this 1 molecule of glucose, we're actually getting a total of 2 molecules out of the citric acid cycle for a total of 4 ATP. But if you look at the formula I gave you, what did I give you as the approximate total number of molecules of ATP that we should get? It's not 4. What should it be? Look at the formula I gave you at the beginning. Right, it should be about 30. Does everybody understand what I'm getting at here? Okay, so we should be able to add them up. And based on the I'm giving you, okay, it should add up to roughly 30, as I'll show you in the end. Okay, for each acetyl-CoA, we get 1. Yeah, it does get confusing. For each acetyl-CoA, we get 1. However, for every molecule of glucose, we don't have 1 acetyl-CoA entering. For every molecule of glucose, we actually have what? 2 acetyl-CoAs entering. Therefore, we get a total of 2 ATP produced in the citric acid cycle related to 1 molecule of glucose. I hope that makes a little bit more sense. Yeah, that's one thing, everybody. I have to keep reminding myself, and I used to do this where I worked before, where I had teaching assistants or TAs. I used to tell them the same thing. I said, be careful, because when you do this long enough, you get used to hearing it, and you forget how it was for you the first time you heard it. So you don't understand why your students are struggling with the information more often than not. But it also, at the same time, what helped me understand this, I had to study a lot, everybody, a hell of a lot, okay? A whole lot. And the definition of working hard was not working hard. You got to go to a whole nother level. So that's the best, I can tell you in tutoring. All right, so you had to know 2 enzymes for glycolysis, hexokinase and pyruvate kinase. I'm going to give you 2 more enzymes you have to know for the citric acid cycle. Okay. Now back here, I forgot to mention, if you look, it shows you there are a total of 8 chemical reactions. There are a total of 8 chemical reactions that make up the citric acid cycle. Okay. The chemical reactions I'm focusing on are the first one and the 6th one. Okay. If you look at the first chemical reaction, the substrate is going to be acetyl-CoA, and the product is going to be citrate. If you look at the sixth chemical reaction, there's a few things I want you to know. One, the product will be succinate. I'm sorry, the substrate is going to be succinate. Again, the substrate is going to be succinate. The product's going to be fumarate, but it's also where we have that second electron carrier coming in. Okay? So what I'm going to show you on the next slide, it's not numbered, but what I'm pointing out to you is the enzyme that catalyzes the first chemical reaction that you have to know, and the enzyme that catalyzes the sixth chemical reaction that you have to know. So we have our citric acid cycle, and we're focusing on enzymes. I changed the font color so it stands out a little bit more once I point it out to you. These are the two enzymes. Now, if you notice something, what do these two enzymes have in common in regards to their name? Hold on real quick. Just answer this question, and then I'll get to it, Jordan. I'm going to actually answer that again, Jordan, in a sec. What do these two enzymes have in common as far as their names go? What do you notice about both their names? Yeah, not 8, but you're close. ACE, right? Okay, yeah, it's about lunchtime. ACE. Well, all the enzymes, notice this. Even this one over here has ACE in it. That's pretty similar to what we saw for glycolysis. You only have to know two enzymes, but they also happen to have names that end in ACE. But when I showed you the other 8, same thing here. Okay. Now, Jordan, I'm going to answer your question because I'm going to begin. Okay. All right. So starting with the first enzyme, citrate synthase. Citrate synthase has an active site that does not complement the shape of pyruvate. Okay. Citrate synthase has an active site that complements the shape of acetyl-CoA. Therefore, citrate synthase catalyzes the first chemical reaction of the citric acid cycle. Therefore, what is the substrate we need to begin the citric acid cycle? Acetyl-CoA, because citrate synthase only recognizes acetyl-CoA as a substrate. Does everybody kind of understand what I mean by acetyl-CoA as a substrate we need to begin the citric acid cycle? It's tied into the enzyme. Okay. Jordan, did you have a question?
Yeah, so just making sure it's the beginning of it that's going to be the product, which is the CO or the citrate synthase or however you say
No,
it.
citrate synthetase is the enzyme that's going to catalyze the first chemical reaction of the citric acid cycle. In order to perform this chemical reaction, it needs a substrate. It's going to convert the substrate acetyl-CoA to the product citrate. That makes sense? Am I answering your question there?
Yeah.
Everybody, you're supposed to have had chemistry. Okay, not to be rude to anybody, but you're supposed to have had chemistry, period. Okay? So if you want to rewrite it out, you can write it out as acetyl-CoA. Underneath it, write substrate. Then arrow pointing away from acetyl-CoA. And on the opposite side of the arrow, or what the arrow is pointing to, you would write citrate. And then underneath the arrow, or above, you would write the enzyme, citrate synthase. That's normally how we do it in biochem, to rewrite it as a chemical reaction. You write acetyl-CoA, then draw an arrow, citrate, okay? Underneath acetyl-CoA, you can put substrate, underneath citrate, put product, and above or below the arrow, write the enzyme that's catalyzing the chemical reaction, okay? That's the best way I can put it. Again, everybody's supposed to have had chem. So listen carefully. This enzyme is the main reason why acetyl-CoA is a substrate required to begin the citric acid cycle. Second enzyme is this one down here, Succinate Dehydrogenase. I forgot I got thrown off there. Listen carefully. If you call for the two enzymes in glycolysis, I try to give you an idea of where the names come up from or derive from. If you look at citrate synthase, It doesn't get its name from the substrate it acts on, acetyl-CoA. It gets its name from the product it synthesizes, citrate. Okay. Down here, the other enzyme, its name is Succinate Dehydrogenase. The first part of its name comes from the substrate it binds to, a molecule called succinate. Okay. Succinate dehydrogenase will strip or oxidize succinate or strip electrons from a succinate, converting it to the product fumarate. Okay. So if you look, succinate, the first part of its name comes from the substrate it binds to. And then what it does to succinate is it oxidizes it. It strips electrons from it, converting it to a product called fumarate. When it strips electrons from succinate, it gives them to FAD, the electron carrier, reducing it to FADH2. Listen carefully, okay? The second part of this enzyme's name, dehydrogenase, in biochemistry, biochemists that tend to name enzymes that take electrons from one molecule and give them to another, biochemists tend to give those enzymes the name dehydrogenase. So dehydrogenase is any enzyme that has dehydrogenase part of its enzyme. That means it's going to oxidize one molecule, take electrons away from it, and reduce another molecule, give electrons to the other molecule. Just look at this part here just to point out what I'm saying. Here's our electron carriers. If I'm not going too blind, I'm missing one here. Here's another one here. Here's another one here. And here's my other one here. I'm not going to test you on these, just listen. This has to do with this one here. This one I will. Notice everywhere you have an oxidized electron carrier entering the citric acid cycle and then exiting the citric acid cycle in a reduced form. Notice they show you these electron carriers by that have as part of their name dehydrogenase, including the one you have to know. So this is in case you ever take biochem. Okay. All right, so again, you can rewrite succinate dehydrogenase. You can write as a chemical formula, writing it left to right. You write succinate, draw an arrow, write fumarate, okay? This time, below the line, below the arrow, you would write succinate dehydrogenase. Above the arrow, you would write FAD, curve line, FADH2 exit. That's normally the way we write it. Okay. So as I said, anytime an electron carrier gains electrons or is reduced in any of the stages of the intermediate stage, it's going to trade those electrons to stage 3. Now, the purpose of glycolysis in the citric acid cycle is to oxidize or strip electrons from glucose or glucose-derived products so that by the time the citric acid cycle, but by the time that 8th and final chemical reaction occurs, there is no product. The molecule's been broken down. There's no product left to be passed on or used or anything. So the citric acid cycle doesn't have a product. It just has a substrate that's needed to begin. We get a little ATP. From one molecule of glucose, we get a net gain of 2 ATP here. We get a total of 2 ATP here because technically speaking, 2 molecules of acetyl-CoA enter the citric acid cycle for every 1 molecule of glucose. But the thing is, I said you get approximately 30 molecules of ATP, so there's still a lot unaccounted for. That's where the third stage comes in, oxidative phosphorylation. Listen carefully. In order to understand or have an idea of what's going on for oxidative phosphorylation, especially the figure I'm going to use, you have to have an idea of where you are, okay? So I'm going to go over the mitochondria. The mitochondria, or mitochondria singular, it's membranous organelle, and I told y'all in chapter 6, so put C chapter 6, membranous organelles. I described a membranous organelle as being an enclosed structure with a membrane or membranes made of the same compounds that form the cell membrane. So let me ask you this question and see if y'all do a better job than the morning class. What is the main organic compound that forms the plasma membrane? Anybody in the class, what is the main organic compound? Phospholipids, exactly. Okay, it's phospholipids. Jordan, again, use the chat board because that's what I see a lot better. I'm real bad with the hand, but go ahead and ask your question.
Oh, okay. So you said membranous organelle is an enclosed structure that what again?
That has membrane made up of the same compounds as the cell or plasma membrane. Just write down C chapter 6. We went over this in chapter 6. Put
Okay.
chapter 6 membranous organelles, okay? All right, so listen
Yes.
carefully. The mitochondria, go ahead and turn your mic off for me, Jordan. Thank you.
Mhm.
Okay, you look here, the mitochondria, it has an outer membrane and it has an inner membrane. These two membranes are not in contact with each other. They're actually separated by an membrane space. What does the prefix inter mean, everybody? Inter. I mean inner inside, but what does inter mean? Yeah, between. So there is a space between the outer and inner membrane, but carefully, there is a solution filling this space. It's not an air-filled space. There is a solution. Okay. Next is I want to focus on is the inner membrane. Outside of the inner membrane, we have the inner membrane space. If we cut the inner membrane away, like we did here, okay, you find another fluid-filled space called matrix. Okay, so both the matrix and the intermembrane spaces are open spaces that are filled with a solution, except the intermembrane space is between the outer and inner membrane. The matrix, okay, is within or bordered by the inner membrane, but they're both fluid-filled solutions. This is important to know as we go over this. So listen carefully. The figure I'm going to use to explain stage 3 magnifies these three areas. The intermembrane space, located outside of the inner membrane, and the matrix, which is enclosed within the inner membrane. That's this figure here. Okay? This right here is the inner membrane. Yeah, the matrix is enclosed by the inner membrane. Okay, so let me go back. I'll give it to you two different ways. Here it shows you on the figure. It's pointing to this open space here. If that's not good enough, that's why I gave you this figure here. Okay, this figure again is pointing out the matrix, and it's pointing right here into this open cavity. And this open cavity is surrounded by the inner membrane. Except what appears to be an open cavity, I said, is filled with a solution. So there's two figures. Now, so this right here is the inner membrane. And notice something, everybody. Does the mitochondrial inner membrane look a lot like the cell or plasma membrane? Yes or no? Okay. So let me ask y'all, does this inner membrane look a lot like the cell or plasma membrane we talked about in chapter 7? And a little bit in 6. Does it look a lot like a cell membrane or plasma membrane? Yes or no? Yeah, it looks very similar. It's a phospholipid bilayer. And it has transmembrane proteins and peripheral proteins associated with it. This is for membranous organelles. You can look at the mitochondria, you can look at the rough ER, the smooth ER, the Golgi apparatus, and when you look at those membranous organelles, what you're going to find, okay, are the same compounds that form the cell or plasma membrane, form the membrane of those membranous organelles, and their membranes look very similar, okay? But I'm not showing you the cell membrane, I'm showing you the membrane of the mitochondria. Notice up here, outside of this inner membrane, this represents the intermembrane space. Okay, what we can't see is the outer membrane would be off the picture here. Think of it that way. Now notice here, I said the inner membrane encloses an area that is filled with a solution called the what? Matrix. Okay, so you got to think of it this way. If you're sitting in a room right now, imagine the walls around you represent the inner membrane. And the open space you're sitting in is the matrix. Except imagine the air being a solution. That's the best way I can describe it, everybody. I can't break it down any more than that, okay? Well, the reason why I'm explaining this, and the main reason why I have this figure, is because what scientists discovered is that the inner membrane has many transmembrane proteins, like I was showing you. And many of these proteins function as what, everybody? Enzymes. And not just enzymes. If you look over here, and you look at the molecules they're showing here, all of these are molecules that are in the formula I gave you for cellular respiration. So this should give you an idea that the enzyme, these inner membrane, has many, many transmembrane proteins in peripheral that function as enzymes involved in cellular respiration. So let's get going here. That was the easy part. Okay, I have the inner membrane here. Now, oxidative phosphorylation, this is the third stage. Okay, and this third stage consists of two sub-stages. The first sub-stage that happens first is a process called electron transport. The second sub-stage, which happens second, is a process called chemiosmosis. This second sub-stage, chemiosmosis, is a process that cannot occur unless the first stage, the process called electron transport, occurs. Okay, so chemiosmosis is dependent on electron transport happening. Okay, that's the first thing. Second, this third stage has a long name, but the name, the two-part name here is associated with the two substages. Third stage is called oxidative phosphorylation. The oxidative here is referring to the first process and what happens to our electron carriers. Well, I just said we have the reduced form of an electron carrier called NADH, transferring electrons from stage 1, 2, and the intermediate step. We have a second electron carrier in its reduced form called FADH2, exiting the second stage and transferring the electrons to stage 3, oxidative phosphorylation. But now what I'm telling you is you have to know exactly where are they transferring these electrons to this third stage. It's to this first process. Listen carefully. Electron transport is a process catalyzed by a network of proteins called the electron transport chain. Okay, so again, these guys right here, all of them that I've boxed in or circled in, this is a network of proteins that collectively as a group they're called the electron transport chain, or simply the ETC. Okay, the ETC forms a process called electron transport. However, they need energy to perform this process. The ETC, this network of proteins, they do not get their energy from ATP. Okay, they get their energy from the flow of electrons going through them. That's what the yellow lines represent. The yellow lines and arrows represent electrons flowing through them. Where are these electrons coming from? They're coming from the electron carriers in their original form, NADH, FADH2. Notice for both electron carriers, okay, this part, oxidative, happens. Both electron carriers, when they arrive here at the ETC, NADH in its reduced form will enter the ETC and exit in an oxidized form. What is happening? It's transferring the electrons to the ETC, and it loses the electrons to the ETC, and now it exits as oxidized form. Yes, they start in their reduced form. Remember what I said. Electron carriers exiting stage 1, 2, and the intermediate step. When they exit those parts of cellular respiration, what form are they in? They're in a reduced form. They're carrying what? It says right here, electrons. However, they're carrying them to stage 3. Okay, no, it's okay. They're carrying them to stage 3. What part of stage 3? The first substage, electron transport. This electron transport is a process, a function, okay, carried out by a group of proteins, this group of proteins that form the electron transport chain, also known as the ETC. But the ETC needs energy to do this work. Where do they get that energy from? From the electrons that these electron carriers have carried over from stage 1, 2, in the intermediate step to stage 3. The oxidative part is in reference to what's going to happen to the electron carriers. They enter stage 3, specifically enter the ETC, and they are oxidized. They lose the electrons they were carrying to the ETC. They exit in an oxidized form, thus the oxidative. Look at FADH2. It enters stage 3, specifically the ETC, in a reduced form, and then it exits the ETC in an oxidized form, thus the oxidative. The electrons they lose, okay, are actually being given to the ETC. As the electrons flow through the electron transport chain, it gives the ETC the energy it needs to function as a proton pump. Okay? So again, these two electron carriers are oxidized, thus the oxidative. They lose their electrons to the ETC, so you can see electrons flowing through or electrons transporting through. And these electrons, as they transport through the ETC, give the ETC the energy it needs to function as a proton pump. Protons are pumped out of the matrix and into the intermembrane space, and this results in a high concentration of protons in the intermembrane space solution and a low concentration of protons in the matrix solution. So you have to understand membrane transport in Chapter 7 to help you with this part of electron transport of oxidative phosphorylation. Let me ask you a question now, feeding off of Chapter 7 and using it for this. Whenever we have a solution on one side of a membrane that has a higher concentration of solids than a solution on the opposite side of the membrane that has a lower concentration of solids, what do we call this area here between the high and the low concentrations? What is it called? It had to do with membrane transport, a passive process. Right, concentration gradient. So what is the ETC actually doing by acting as a proton pump when it has the energy it needs from electrons flowing through it? It's creating a proton concentration gradient. This is important for the second substage to occur, okay? Now, we have two reactants that are needed for cellular respiration, right? We need glucose. It is the very first substrate used in the very first stage of the very first chemical reaction for glycolysis. Okay, so glucose is not just a reactant, it's a substrate. But O2, it's not a substrate, but it is a reactant. But I said more specifically, what role or function or purpose does O2 have in cellular respiration? What did I tell y'all last class? What is the role it plays? What is its function? I know I gave it to y'all. Doesn't take hydrogen at the end. Glucose is a substrate needed to begin glycolysis. Okay, don't get fooled by that. Right, there you go. We're going to get one of them. Thanks, Nicholas. Yeah. So in other words, the electrons that were being stripped from glucose and glucose-derived products when they were being oxidized, and those electrons that were then carried over here to the ETC so that the electrons could flow through it, where do those electrons that initially came from glucose and glucose-derived products end up at finally? Well, they end up being accepted by the final electron acceptor. Who is our final electron acceptor? O2. So the electrons that flow through the ETC, the electron transport chain, this network of proteins, the energy they need to pump protons, creating a proton concentration gradient. But the electrons will flow out of the electron transport chain, where who is there waiting to finally accept those electrons? O2. And what happens when O2 accepts those electrons? It's going to be H2O. Now, is there more to the story? Yeah, there is, but I'm not going to cover that here. Okay, so I understand the protons, but focus on what I'm giving you for now. You take biochem, if you have a good biochem professor, they'll definitely fill in the details. Right now, I'm just trying to hope. I'm hoping that when you leave the class, you're better off than when you came in, to where when you take your higher level courses, your upper level, that you have a better foundation. Even if you don't remember something word for word, hopefully you'll recognize it much quicker, and then it'll start to come back to you, but you'll understand it even better. That's what I'm hoping for. Okay, so when does electron transport come to an end based on the way I'm talking about it? When the electrons finally flow out and O2 accepts the electrons, because it's the final electron acceptor, converting it to H2O. What do we have created? A proton concentration gradient. Now, the second substage can occur, chemiosmosis. This may not be word for word what I gave you in your notes, okay, but chemiosmosis is defined as work done in the presence of a concentration gradient, or using a concentration gradient. Okay, well, specifically not a concentration gradient. Chemiosmosis is work done in the presence of a proton concentration gradient. Well, for chemiosmosis, I first say it's work done. Well, what type of compounds do work in a cell, everybody? I've asked this question a lot. Of the 4 organic compounds, which of the 4 did I say does the work in a cell? There are 10 of you here. I talked about several of them. Of the 4 organic compounds, not phosphates, of the 4 compounds, proteins, yes, proteins. Proteins do the work in a cell. Well, what kind of proteins have we been focusing on in this chapter? Proteins that function as enzymes. They catalyze chemical reactions. So that definition, I know I didn't give it to you word for word, but that definition I gave you where it says work, well, what's doing the work is a protein enzyme called ATP synthase. It's the one performing chemiosmosis. It's the one doing work in the presence of a proton concentration gradient. Now, how is it able to do this work? Well, it just so happened that scientists discovered not only is this enzyme ATP synthase a transmembrane protein located in the inner membrane of the mitochondria, but they found that this enzyme, okay, doesn't get energy from ATP, doesn't get energy from the flow of electrons. It gets energy from the flow or the diffusion of protons from a high concentration through a channel in this enzyme so that protons can diffuse from a high concentration in the direction of the low concentration of protons. So ATP synthase, technically, it's a channel. It allows protons to diffuse from a high to a low concentration of protons in the matrix. What scientists discovered is as these protons diffuse through ATP synthase, it gives the enzyme ATP synthase the energy it needs to synthesize ATP. So notice they're showing you how does ATP synthase synthesize ATP? It takes AD, adenosine diphosphate, and it covalently bonds a phosphate group, creating ATP. Well, I went over that. Is that an example of phosphorylation? Is the addition of a phosphate group to a molecule, is that phosphorylation? Yes or no? Ten of you here and nobody has an answer? Yeah. 50-50 chance. What do you got to lose? Guess. Yes, it is. Okay. So in other words, this enzyme performs a process called phosphorylation. Okay. It phosphorylates ADP. It adds a third phosphate group through the formation of a covalent bond. However, when this covalent bond is formed, attaching this third phosphate group to the second phosphate group of ADP, producing this energy molecule called ATP. What do we call that type of energy chemical reaction or energy associated chemical reaction? There were two types we talked about in chapter 8 that are energy associated chemical reactions. One of the two produces a product rich in energy. Because when the covalent bond is made, energy is being absorbed. What do we call that type of chemical reaction? Close, not endothermic, but the endo's right. Yeah, endergonic, endergonic. Thermic, we're talking about body heat, okay? Endo and ectothermic, that's cold-blooded, warm-blooded. Okay, we're talking in this case endergonic versus exergonic. So in other words, ATP synthase is performing phosphorylation, but since this type of phosphorylation is resulting in the production of a product rich in energy, it's an endo ATP synthase. That's what it's catalyzing. But where is it getting the energy that it needs? Not from ATP, not from the flow of electrons, from the flow of, or from the diffusion, okay, or the movement of protons from a high concentration through it to a low. Now, technically speaking, okay, I want you to just go, imagine it as just going in this direction in the test, okay, but in reality, this is a cyclical process when it comes to the protons. The protons will diffuse back to the matrix, but the ETC, as long as electrons are continuously brought to it or carried to it, will pump them right back out. So in other words, the ETC has to ensure it maintains this proton concentration gradient so that protons continue to diffuse through ATP synthase so that it doesn't do this one time, but continuously synthesizes what? ATP. So these protons are constantly being cycled around. That's the reality of it. For the test, just imagine we're going in one direction because that's hard enough for right now. Okay? Any questions on this part? What about you, Jordan? You got quiet. I didn't see no hand up. Did you leave? Okay. Okay. So this is oxidative phosphorylation. The names are tied into these. Okay. Yeah, it's getting hard. I say it in a nice way. I told you at the beginning of the semester that beginning with chapter 2, okay, the information builds on itself. Some of my students automatically understand that means it's going to get more difficult, so I better make sure that I learn the information early on. Because the better job they do, then the less difficult it'll be later. Okay? So Some of the areas you need to review for this part, for cellular respiration, you need to go back to chapter 8. You need to go back to chapter 7. Okay, you even need to look at organic compounds too. So I don't give a comprehensive exam for the final test at the end of the semester because I shouldn't have to. And this is the way your biology classes should be. Now, some professors will give a final exam only to help you manipulate your grade. That doesn't mean you learned anything. Okay, I'm just not that way. Okay, I actually this information. So if you look here, the formula I gave for cellular respiration was 1 molecule of glucose plus 6 molecules of O2, which will play a role down here, will result in the production of 6 molecules of CO2, 6 molecules of H2O, and the squiggly line here means approximately or about, in this case, 30 to 32 molecules of ATP. I put 30 to 38 up here because it depends on the textbook you see. And every once in a while, I do get students in this class that actually have not only a strong chem background, they have a pretty strong biochem background. Okay, so but what I gave you for the formula at the beginning of the PowerPoint and in your notes is what I'll use for the test. Okay, so I said we were tracking how much ATP is produced from 1 molecule of glucose. We get a net gain of 2 ATP in glycolysis. We get a net gain, or not a net gain, but we get 2 molecules of ATP produced. I don't say net gain because none was required for the citric acid cycle. And then for chemiosmosis, ATP synthase will synthesize approximately 26 to 28 molecules of ATP, maybe a little more, again, depending on the textbook you're looking at. But notice something. The vast majority of ATP is produced at the end. Okay. What researchers and scientists discovered is that if O2 is not present to function as the final electron acceptor, okay, it cuts off this process. Electron transport doesn't occur. Now, I haven't looked into why. I don't know if anybody actually knows why, but there are insecticides, there are antifungal creams, and even the poisonous gas carbon monoxide and the poison cyanide, they all interfere here. And what happens to the living organism? It dies. So the only time cellular respiration is not occurring is if the cell or organism is dead, everybody. This is a constant process, okay? The ATP is being used as fast as it's being made. It needs to be the final acceptor, yes. Yeah. Listen, side note, for carbon monoxide, what researchers discovered is that your red blood cells actually, the hemoglobin, actually does a better job of binding to carbon monoxide than O2. So what ends up happening is when you inhale, if there's carbon monoxide in the air, you're inhaling oxygen, O2, but you're inhaling carbon dioxide and your red blood cells decide more of that. So what ends up happening is your cells don't have enough of this to act as a final electron acceptor. So now what happens is the majority of ATP the cell can't produce through cellular respiration because electron transport has been inhibited. So what happens to the organism overall? It needs a lot of ATP just to stay alive constantly, continuously. So the organism dies. I'm not going to test you on this, but this is just to let you know. Fats and proteins are fine, specifically fatty acids in the backbone glycerol, proteins, specifically amino. But long-term is not good, such as for type 1 diabetics or somebody who becomes type 2, chronic conditions to where 10, 20, 30 years pass. The breakdown products, the waste products produced using these two long-term, 10, 20, 30 years can have very negative effects on the body. Our cells of our body, and especially our nervous system, is pretty exclusive. Prefer to use carbohydrates or sugars, specifically glucose. So it's not that other nutrients that can't be used. It's just glucose can be used as a clean source of fuel if you want it. Our body can handle any waste from it and control it much better long term. Short term, our body will intentionally, okay, go into a process that they sometimes call glucose conservation. This is where, with the exception of our nervous system, many, if not most of the cells of the rest of our body will be told, hey, go ahead and use the fats, specifically fatty acids, and the proteins, specifically amino acids, to make ATP for a little while. And it's fine. It's fine. And then your body will tell those cells, hey, switch back to the sugars. So I'm not going to test you on that, give you insight. If you take AMP, they should go over it. Okay, they should go over that. Yeah, we start getting into that. Yeah. Yeah, as far as I know, at least, yeah, they do go over it. Is it AMP2? I think. If maybe they might touch on an AMP1. Definitely by AMP2, it should be a lot more course sentences. And if you're going into the nursing program, we'll definitely go over it. Guarantee it. One of the nursing students that was an hourly for us at one of the labs, biology labs, I remember her final semester, she was writing a paper on ketoacidosis and diabetes. So just to let you know, so I'm not going to test you on what I talked about with this slide or the information on here. It's just a little tidbit for you in case. Yeah, Intro to AMP, it's an intro course in relation to AMP 1 and 2. It's definitely an intro course. So yeah, I taught that course, 2404 is what it's titled, and even I touched on it briefly. It's AMP 1 and 2, one of those two, I believe it's 2. Yeah, they'll give you more, definitely they should. Oh, I don't know. A lot of students are complaining. That's why I got away from it. But well, let's see how we can go from here, okay? All right, so listen carefully. Cellular respiration. Oh, did they? No, 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 no. I wish we were in person, everybody. I really stress the labs. And you hate me or you love me. And the students that are serious, they say don't change a thing. Because I really want you not only to just show up to lab, I want you to learn how to use the equipment. I want you to understand what's going on. Even if it's not perfect, I just want you coming out better than what you came in. So, but yeah, I can't do anything about that. Thank you. Okay, let's move on here. Now, cellular respiration, I say collectively, it is an aerobic process. It falls under aerobic metabolism because we need O2 to perform this entire process. But then it says, what if O2 is not available? Well, there are organisms that are referred to as strict anaerobes. Okay, chemical reactions in them never require O2, and they actually are sensitive to O2. It's a poison to them. Then there are organisms, as well as cells in the human body, that can actually transition. They can perform cellular respiration to produce ATP, but if oxygen is not present, these organisms or cells of the human body can switch over and perform fermentation, which is an anaerobic form of ATP production. In other words, it falls under anaerobic metabolism. Now the catch is you only get a fraction the amount of ATP produced compared to cellular respiration. Okay, but yeast are known to be able to perform cellular respiration as well as fermentation. In the human body, our skeletal muscle cells that form organs called skeletal muscles have the ability to switch from cellular respiration to fermentation whenever you're exercising to the point of, say, fatigue. But it only lasts a short while, okay? So what we're going to talk about is fermentation. Now, just like with cellular respiration, the purpose is to produce ATP. But now there's no O2 involved. So it's a type of anaerobic metabolism. Now, there are two types of fermentation: alcohol fermentation and lactic acid fermentation. Alcohol fermentation is also known as ethanol fermentation because that's the specific type of alcohol being produced, ETOH. I won't use the abbreviation on the test. I'll use alcohol or ethanol, but just to let you know. Lactic acid is sometimes called lactate. I try to use lactic acid fermentation because I just feel like it sticks better. But these are two types of fermentation. Okay. Now, if you notice, okay, for fermentation in general, it doesn't matter which type of fermentation we're talking about. Just like cellular respiration, fermentation involves a stage called glycolysis. Like cellular respiration, okay, fermentation consists of more than just glycolysis. There are additional chemical reactions outside of glycolysis. However, because fermentation, just like cellular respiration, begins with glycolysis, what is the substrate that we need to begin fermentation? Glucose. Okay. Now, the first type of fermentation, it is known as alcohol or ethanol fermentation because of the final product it produces using the substrate glucose. Okay? So alcohol fermentation, it begins with the substrate glucose, okay, and a series of chemical reactions that we refer to as glycolysis. Therefore, at the end of glycolysis, glucose is still converted to 2 molecules of pyruvate, and that is happening to get molecules of ATP produced. Except outside of glycolysis, there are additional chemical reactions that occur. One of the chemical reactions results in the oxidation of pyruvate and the release of CO2, producing a different product that I will not test you on. Okay? And then the other product, it is acted on by enzymes to form the third and final product of alcohol or ethanol fermentation, which is ethanol. Okay? So this is alcohol fermentation. It begins with the substrate glucose. It ends with the final product, ethanol. In between that is a series of chemical reactions called glycolysis, where we get a net gain of 2 molecules of ATP. And then also in between that, we get a couple of molecules of CO2 produced. Okay. Our skeletal muscle cells do not perform this type of fermentation. However, living organisms called yeast, which are types of single-cell unicellular fungi, do perform this type of fermentation. Now, they can switch over to cellular, but they can perform alcohol fermentation. Okay. The other type of fermentation, this type of fermentation is performed by our skeletal muscles. Listen carefully. I put lactate here, but the lactate should be here in parentheses. Okay. You take biochem, they'll tell you, hey, pyruvic acid can be called pyruvate at certain times. Acetic acid can be called acetate at certain times, and lactic acid can be called lactate at certain times. I leave that for biochemists, okay? Second type of fermentation, it should say lactic acid and then lactate in parentheses, then fermentation. This is performed by our skeletal muscle cells, so I'm not going to write it out. You can write it out. Our skeletal muscle cells perform this second type of fermentation when you're exercising to the point of fatigue, okay? But just like the first type of fermentation, alcohol fermentation, it begins with the substrate glucose, and it includes a series of chemical reactions called glycolysis. However, there are additional chemical reactions that occur outside of glycolysis, okay? But when glucose enters this series of chemical reactions that we call collectively glycolysis, we get 2 molecules of ATP being produced, as over here. And we end up with 2 molecules of pyruvate produced. However, not shown here is there are enzymes in our skeletal muscles that will then act on pyruvate as a substrate and convert it to the final product of this type of fermentation called lactic acid or lactate. Okay, so this is the second type of fermentation performed by our skeletal muscles. Now again, Notice it's a lot less ATP. We're just getting 2 molecules of ATP for every 1 molecule of glucose for these 2 types of fermentation. Okay, it doesn't matter which type it is, we're getting very little ATP. Okay, so either the organism has to be unbelievably tiny that it can get away with this and not need so much energy, or in the case of our skeletal muscles, this is only for a brief moment. It's not going to last long, okay? All right. Listen carefully. This is the last slide. Make sure you make a note. I'm not going to test you on this, but it's just in case you go on. A lot of my students go on a micro. There are organisms referred to as obligate anaerobes. Okay, these are what we also call strict anaerobes. Okay, they carry out fermentation to produce ATP. Okay, and O2 is actually poisonous to them. They cannot survive. This is what you see primarily for prokaryotic organisms. As a matter of fact, the first living organisms that evolved are believed to have been strict anaerobes because there wasn't enough, the belief is there wasn't enough O2 in the atmosphere to support aerobic metabolism. Okay, and some of those still exist today. They're referred to as obligate or strict anaerobes. However, there are those such as yeast, unicellular fungi, some bacteria, or in the case of our bodies, our skeletal muscle cells, that are facultative. That means they can perform both fermentation and cellular respiration, depending on whether or not O2 is available. Okay, but if they perform fermentation, more often than not, it's not something they want to do long term. Yes, I know it gets brutal. It's a lot of biochem, everybody. It's a lot of biochem, and you'll get even more for photosynthesis. I'm going to stop the recording here.